Okay, so the microphone is working. That's excellent. So I'd like to start off by saying thank you to Thomas and Klaus for the invitation to be here. It's a real pleasure. Mein Deutsche ist nicht gut, so I'm very pleased I can talk in English. And yeah, we have the excellent translators at the back who can translate it for you. I apologize in advance for any English humor. I won't be offended if you don't laugh. Okay, so today, well, we've heard some really excellent talks from developers of these amazing AI tools. I haven't developed any of these AI tools myself. I'm a scientific user of these tools, and so I want to tell you a story about how really these tools are revolutionizing the work that I do. And so thank you to all the developers for your amazing work. So I'm, as we've heard from Ulla, I'm part of the European Molecular Biology Laboratory, which is headquartered in Heidelberg. But I'm based at the outstation near Cambridge called the European Bioinformatics Institute. And so what we produce there are data resources. And at EBI, we have 800 staff. We produce 40, or more than 40 different data resources across a range of, across a range of different parts of the life sciences through, chemistry and drugs, genes, DNA, proteins, cells, organisms, the whole range of things. I'm sort of focused on a more narrow part of that, and so here in blue circles we have the different data resources that I'm in charge of. So at the top we have UniProt. Can I get a mouse? Okay, seems I can't drive it very well. Here we go. So UniProt is a large collaboration between partners in Switzerland, the US, and the UK, and we produce a large database of protein sequences and the annotation, the knowledge about them. We have resources for non-coding RNAs, RFAM and RNA Central, but today I'm going to be talking about the protein part of it down here, so mainly PFAM, and I'll maybe mention Interpro. Okay, so let's change fields entirely for a moment. Let's go to the famous Kiwi physicist, Ernest Rutherford. He famously said, all science is either physics or, and does anyone know the rest of this quote? Louder. Okay, it was, I'm not sure if anyone got that, I couldn't hear. Stamp collecting. And this was really aimed at biology, which he didn't feel was much for real science, and we were just gathering a bunch of random facts together. But, I think scientific stamp collecting is extremely important. I spent the last 30 years of my life doing this. And a really good illustration of why scientific stamp collecting is important is if we move now to chemistry. And so Mendeleev came up with the periodic table of elements, and this allowed him to organize what was known at the time, understand the properties of the different atomic elements, organize them, and also make predictions. So he was able to predict the existence of gallium and germanium and some of their properties many years before they were isolated in the lab. Now, of course, chemistry is pretty simple, right? If we move on to something a bit more complicated, like all life on the planet, well, how do we systematize, how do we stamp collect here? And so enter Carl Linnaeus. And so he gave us this systematic way of organizing all life on the planet. He gave us the binomial nomenclature of species, Escherichia coli, Homo sapiens, and so on. And we see him here with one of his collections of leaf morphology. So now, Carl Linnaeus, what he didn't understand was how did this diversity of life on the planet arise? So enter Charles Darwin, who gave us the theory of evolution by natural selection. And so he could explain how new species could arise from pre-existing species by mutation and selection. So, now, what I think neither Linnaeus nor Darwin understood was what I believe is the real fundamental complexity of biology. And to see that, we can zoom in, and here we see a bacterial E. coli cell, or a rendition of it. And it's just full of all of these molecular machines that make life possible. And so my stamp collection is going to be to try to understand all of these protein machines, what they look like, and what they do. And so I'll move on to the classification of proteins. This work was really motivated by a paper that my PhD supervisor published, which was literally a back-of-the-envelope calculation, which was published in Nature. I wish it was quite as easy to do that these days. And he said that the large majority of proteins come from no more than 1,000 families. And I'd spent my entire life collecting things, records, fossils, stamps, all sorts of things. So 1,000 things, that didn't seem like so many to collect to me. So after my PhD, well, actually during my PhD when my supervisor wasn't looking, I started to classify proteins into families. After my PhD, I went to work with Richard Durbin on this database that had been newly founded called PFAM, for protein families. And in the last 3 decades, my personal scientific goal has crystallized to classify all proteins completely and accurately. So let's have a look at a protein. So here we see the structure of phospholipase C, which is an enzyme that cleaves membranes, and it's from a nasty bug that causes gas gangrene. And so here we can see the structural view in the top left, and in the bottom we can see the simplified view. And the protein is made up of what we call domains. So it's got 2 domains, this one made of alpha helices and this one made of beta strands. And nature recombines these domains. They're like Lego bricks. It can put them together in different ways. So the alpha helical domain on the left is the enzymatic domain that cleaves phospholipids. The domain on the right, we're not 100% sure, but we believe it localizes the proteins to membranes. So as you can see, it's quite easy to spot where the domains are when we look at the structure, but actually, for many, many years, we haven't been looking at the structures because we didn't have them. We were looking at the sequence of the proteins. And what we would do is we would make multiple sequence alignments, so each row here is a different protein, line them up, and then we can see which parts of the protein are conserved. These are sort of the light blue colors. We can then use statistical models, a machine learning technique called a hidden Markov model, to model that, and then we can go and search in any new genome that's sequenced and find examples of that protein family. So in these last 30 years, me and my team, we've collected together 24,000 different protein families. There are many still to be added. And importantly, we add annotation. We go read the literature and try and extract the knowledge for each of these families and write annotations so that the users of our resources can understand what's going on. And so a new release of PFAM is coming soon. It will have 25,000 families in it. Fantastic. 76% of known proteins are matched by this database. So we've already heard multiple times about AI-based structure prediction. And so the Nobel Prize last year went to Demis Hassabis and John Jumper from Google DeepMind, and David Baker as well, who was an academic who worked in this field too. And they produced this tool called AlphaFold. And I understand AlphaFold quite well, so let me give you the one-slide summary of AlphaFold. Sequence goes in, something, structure comes out. This puts me in mind of a cartoon by Sidney Harris. And I wish I had time to explain what's going on in the black box. I don't, but I'd be happy to talk to anyone afterwards about that. And so this tool has now been applied by Google DeepMind, and they started to predict the structures of many proteins. They started off with 21 model organisms and predicted all of those. In a meeting before AlphaFold was released publicly that EBI was having with John Jumper and Demis Hassabis, I remember Demis saying, well, Alex, what would you think if we could predict the structures of everything in UniProt? And, well, I said, this would make my dreams come true. And within 1 year, this is exactly what we had. So the next release of this AlphaFold database had essentially all the proteins in UniProt, or something that was very closely related. We went absolutely wild with all of these structures. We clustered them. We could find all sorts of new, interesting, very pretty looking kinds of structures in there. We still don't know what they do, but they look fantastic. And so the next step will be, trying to determine what they all do. Now that we have structure prediction, this makes protein classification actually quite easy. Remember, I said if we have the structure, we can see where the domains are. So here's a family I built in 2004. It's called calmodulin_bind. And here's the multiple sequence alignment. We knew it was found in plants. We thought at the time it was involved in stress response. There was never an experimental structure of this. But then, suddenly we have an AlphaFold model. Oh, it's got 3 domains in it. And so we can immediately go back, chop up our family into the 3 bits. Because we have structures, actually, we can go and search the database of known structures with each of these domains. And what we find is that domain 1, this thing over here, it's actually similar to a known DNA binding domain. And so it turns out these proteins are plant transcription factors. So then last year, the Google DeepMind group, they produced AlphaFold 3. And so whereas AlphaFold 2 could predict the structure of a protein from the sequence, and that was it, now AlphaFold 3 could predict the structure of the things that bind, the non-protein things that bind to the protein, like DNA. And so we could then ask AlphaFold 3, can you show me what this looks like with DNA? And so we can see the first domain is binding to the DNA as expected, but actually the DNA binds between the first and the second domain. This is just incredible. So AlphaFold is allowing us not only to understand the structure, but now to explore the functions of these proteins. So in the example where I was showing that we were looking at the whole universe of AlphaFold structures, we were building new families from that data. And one of them is unimaginatively called HTH_73. This stands for helix turn helix 73. And this is a known class of DNA binding protein. But now with AlphaFold, well, we can go ahead, we can find out that actually these proteins bind as dimers, that they do bind to DNA. And here's a 17-nucleotide DNA sequence that my daughter kindly designed for me that has all the dinucleotides in it. Great. And the yellow color here means it's not very confidently bound. So what if we could try every possible DNA sequence and see which one bound best? Would that be what this binds to in nature? Of course, it would be very expensive. This is, even if it was just 10 nucleotides long to try 10 to the power of 4 combinations, that's like over 1 million different combinations. Too expensive. But I had sort of a flash of inspiration, and I don't know how many of you here have ever tried to crack open a combination lock. Okay, yeah, okay, at least one person. So the way you do this is you don't go through all 10,000 combinations. You hold it and you sort of pull, and you go through one combination at a time until it clicks and you can feel a bit more movement. And then you move to the next position. We can do this with DNA. So we'll just change one position at a time, find the best one, move to the next position, find the best one. Much more efficient. And so when we do this, we get an extremely confident model. We get a sequence. It may not be the perfect binding site for this protein, but I think it's very likely to bind. And if you have an experimental lab and want to test this, be very happy to see. Also, we were able to go a step further because we knew from other evidence that this protein was involved in restriction modification, which is a bacterial virus defense system. We guessed that this might bind to methylated DNA. And in fact, when we methylate the cytosines, it binds even more strongly. So, yes, AlphaFold, it's changing my scientific life and what we can do and the speed at which we can study proteins. Now, we've heard a lot about large language models as well. So perhaps I can say something about how we get ChatGPT and Claude to do our homework for us. So one of the things I said was that for each of these families, we have to write annotation. We have to read the literature. This is very laborious and slow. And takes a lot of time. And so here we see a view of one of the web pages showing one of these families, and you see quite detailed annotation is written. If you just ask ChatGPT to write the annotation for this family, if it's a very well-known family, it can do something, but it doesn't give you sort of correct references, all sorts of problems. But what we can do is if we feed it the right information, and we take that from UniProt, the well-curated parts of UniProt, and then we ask ChatGPT to summarize that, it does a spectacularly good job. I mean, really very good. In fact, we tested a blind test of our curators with the annotation they'd written versus the annotation that ChatGPT writes. They prefer the ChatGPT annotation. So in this one, we're sort of giving chunks of text. It's very well organized. Can you go further? Well, actually, and I'm a Claude user. I love Claude very much. And here, I just say to Claude, well, here's a PDF of a paper. Read the paper and write the desk file. That's the annotation file for the GP28 protein. And it goes off and it thinks about it, and it writes this very well-structured annotation. And it's better than what I would have written, I can assure you. So I think there are huge opportunities. These models, if you treat them well and you explain very clearly what you want, they can deliver amazing results. And just to say, this isn't all the information that I gave to ChatGPT. This is in something called a project. Actually, I've already told it all of the standard operating procedure for PFAM and how to curate families. There's a lot more information behind this to get good information. So, how has this changed what we're doing in this PFAM database? Well, let me show you how many new entries we've added each year over the last couple of decades. And so on the x-axis, we have the year going from 2004 up to 2025, so yeah, 21 years. This is the number of new families going from 0. Here's 1,000, and that's, on a good year in the past, we've been able to build 1,000 new families. And that's why over sort of 30 years, we've built 25,000 families. But you can see things are changing as these models have come in. And this year, I'm very confident. I keep telling my team this, that they're going to build 4,000 families this year. And I believe that they will manage that. So, of course, that's not enough. As we've heard about AI agents, I believe that they will become really quite useful and practical next year. And then, so I think we'll be able to build maybe 6,000, 8,000 families, and beyond that, who knows? So, actually, now I think, although I thought that honestly, that we would never complete this classification, that I would probably retire before it was done. Now, I'm absolutely confident that we will have something that's very close to a complete classification, maybe some inaccuracies, maybe it won't be complete, but it'll have all the important stuff in, and we'll get that done by 2030, which I'm young enough that I still have 10 years before retirement, leaves me 5 years to then go and work out what all these proteins are doing. Okay, so let's just wrap things up there. So, yeah, my life's work has been to create this complete and accurate classification of all proteins. And, yeah, it's been a significant challenge, a lot of work, but we've got there in steps. AlphaFold coming along it's just like we've been fumbling around in a dark room, trying to build these families for 20 years, and someone has now switched the lights on. I can just see, I look at the protein, there they all are. And so now we just have to classify them. The large language models are meaning that actually that part about reading the literature has become not quite trivial, but much, much easier and faster and more accurate. But I still believe there are huge opportunities in unlocking all that knowledge that's in PDF files, in PubMed and so on, and actually extracting the knowledge from that and organizing it into data resources. So yes, my bold claim is by 2030, PFAM will be finished, and hopefully I've explained to you that this is why AlphaFold and AI are changing my life. And I'd like to finish by thanking the team. There's a lot of people who are involved in doing this, and for all the different people who are funding this. And finally, I'd like to thank you for your attention. Thank you.